So Patrick Fisher is a senior researcher at the Neurobiology Research Unit in Copenhagen, Denmark. He has a PhD in neuroscience from the University of Pittsburgh in the USA. He supervises many of the functional brain imaging research projects and clinical collaborations ongoing in his lab. Over the past 6 years, he has supervised the psychedelic brain imaging studies that apply molecular and functional brain imaging to resolve acute and lasting effects of psychedelics. Some relevant clinical collaboration of this group is the Connect Me project led by Dr. Danielle Gonziella. That probes functional brain imaging markers of consciousness in patients with disorders of consciousness. So thanks a lot, Patrick, for being here. And whenever you're ready, the floor is yours.
Yeah, great. Thanks so much. So thank you, everybody, for joining. It's really nice to be able to talk to you guys and share with you some of the research that we're doing. Unfortunately, we'll do it on the internet, but hey, that's better than nothing for sure. So what I'll talk to you guys about today is I'll share with you a little bit of the research that we've been doing over the last couple of years using human brain imaging to try to explore and evaluate molecular mechanisms and functional mechanisms associated with psychedelic effects in the human brain. I changed the title slightly to include psilocybin here because at least what I'll talk about from the studies that we've done have been exclusively with psilocybin, although I'll talk a little bit about some of the other research that's been done and some of the other compounds that have been evaluated. So yeah, I guess we'll wait for questions. We'll do questions at the end, but I'm super excited to answer any questions that you guys have. So please let me know, and yeah, I'll plan to be in this 40-minute time window. So maybe I'll just shrink your guys' faces right now so I can see all the slides. Let's see. Yes. So the start of what I want to talk about is sort of a broad perspective on why we're interested in psychedelics, or why people are generally interested in psychedelics. This is perhaps self-evident to some of you, but maybe not for others. So just indulge me for a moment. One of the things is that over the last 5 to 10 years, there's been really a booming interest in psychedelics and their effects. Most prominently, this has been, or very prominently, this has been in the popular media, where you've seen a lot of different research articles or let's say secondary articles about recent either clinical studies or human brain imaging studies, some of which have had some quite sensational titles associated with them that are really articulating how psychedelics seem to work and really changing how they might change the way that we think about treating patients with various types of disorders. And then this last bit here this is just a book that was written a few years ago now, by a guy named Michael Pollan. I would recommend this book. I think it's got some really good historical perspectives on the lifetime of psychedelics in Western culture and Western medicine. There's also a little bit of an insight on the neuroscience and human brain imaging of psychedelics in this book. It's on the relatively older end, which is to say it's only about 3 or 4 years old, but there's been a lot of change in the field in that time window. But nevertheless, I think quite a good book. So the main short of it is that there's a very broad interest in psychedelics from a sort of popular perspective. Before I go further into sort of why that interest has emerged, I want to just establish a framework for what we're talking about when we talk about psychedelics, because this can actually vary quite a bit depending on who you're talking to. So I'll first just kind of operationally define what I'm going to refer to as psychedelics, or more broadly as serotonergic psychedelics. So this is a class of compounds that understandably, given the name, have a similarity to serotonin. So here you can see the molecular structure of serotonin. And can I ask, actually, you guys can see my mouse on the screen? Is this right? Yeah,
yes.
good. Okay, great. Thank you. So this is a molecular structure of serotonin. Here we have a cartoon of a serotonergic synapse, right? The serotonin system is characterized by a number of different receptor mediating systems. There are postsynaptic receptor systems as well as autoreceptor mechanisms. The serotonergic psychedelics are a class of drugs that share structural homology or structural similarity to that of serotonin. And you can see that sort of indolamine backbone in each of these compounds. Even in LSD, if you kind of trace this up, you can see the general structure. So these are molecules that have similar structure to that of serotonin. And for those of you not familiar, psilocybin is the psychedelic component in magic mushrooms. It's dephosphorylated into psilocin, which is the active metabolite. And then DMT is the psychoactive component, psychedelic component in ayahuasca, which is a compound, or which is a drink mixed from brewing two different types of plants together, one which includes NMDMT, the other which includes an MAOA inhibitor. LSD, lysergic acid diethylamide, is a synthetic compound. It was synthesized by Albert Hofmann, who's pictured here, in 1938. He then ingested it in 1943 and had a heck of an experience in doing so. But it's the one synthetic compound out of the set. These are all compounds that have their primary psychedelic effects mediated via the serotonin 2A receptor, which I've highlighted here, and showing here a surface rendering of the distribution of serotonin 2A receptors throughout the human brain, which we estimated across a broad population of people with positron emission tomography. And I'll come back to this. This imaging tool is a strategy. I want to just talk a little bit about the pharmacology of serotonin psychedelic drugs. So again, these are compounds that have their primary mechanism of action, or at least the psychedelic element, via modulation of the serotonin system. So again, we are reminded of the structural homology with serotonin, and they're acting via the serotonin 2A receptor. Why is it that we know that the serotonin 2A receptor seems to be particularly relevant for the psychedelic experience? Well, this is because if we pre-administer a drug that blocks serotonin 2A signaling, so a serotonin 2A antagonist, ketanserin, or what some people argue is an inverse agonist, if you pre-treat with ketanserin and then administer a psychedelic to participants, it effectively abolishes the entire psychedelic experience. And that's articulated in these two plots here. On the top, you can see this K2 plus psilocybin, the right-hand bars. These are the administration of psilocybin after receiving ketanserin, and you can see the psychedelic experience is nearly completely reduced. This effect has been replicated a couple times, primarily out of this group in Zurich, Switzerland, led by Franz Wollenweider and also Ketrin Preller. And the plot down here is just looking at a couple different dimensions on the same self-report questionnaire, this so-called altered states of consciousness questionnaire, which can be divided up into a couple different factors. And here you can see in both plots, the effects are, the psychedelic effects are essentially abolished when you administer an antagonist at the 2A receptor. And there's a lot more detailed information that supports the central relevance of the 2A receptor for the acute psychedelic experience that I'm not going to go into now, but suffice it to say it's there. Another thing I want to point out is that while the 2A receptor seems to be critical for the acute psychedelic experience, these are drugs that have a very diverse pharmacology. And I think that's really important to remember. It alludes to some of what we really don't know about how these drugs work. So this is a table from a review paper now about 10 years ago with estimated affinities of psilocin, the active metabolite of psilocybin, at various serotonin receptor targets, as well as other receptor targets. Now, what you can see is there's clearly affinity for the serotonin 2A receptor, and this estimated range of about 100 nanomolar, but you can also see that there are it hits nearly every other serotonin receptor to some degree. You can see it's got some off-site binding, but it's really, there's a polypharmacy associated with these compounds. This is even more so the case with LSD. So here you have this is a tree from a review paper published earlier this year from Brian Roth and a colleague, where they show all of the mammalian G protein coupled receptors, if I remember this figure right. And the green dot that you see down here at the bottom is the serotonin 2A receptor, and the red dots articulate other receptors for which there's an estimated affinity of LSD for that target. Just to emphasize that, again, the 2A receptor seems to be critically important for the psychedelic experience. I'm going to talk a lot more about the 2A receptor as we're going along, but I think it's really important to keep in mind that these are drugs that are acting at multiple targets. LSD, as I just mentioned, hits a number of dopamine receptors, which actually distinguishes it from psilocybin. So I think that's just kind of an important thing to keep in mind about the pharmacology of these compounds. Why are people interested in these compounds? Why are people writing these? Obviously the acute psychedelic experience is very profound. It's got these characteristics, this idea of ego death, this noetic quality, this sense that when you take these compounds, people describe a sense of knowingness about sort of fundamental elements of the universe. People talk about a oneness with the universe, very emotional experiences people can describe, sometimes very challenging experiences. Certainly that's quite compelling, but in terms of doing research on these compounds, there really needs to be a broader relevance. And this is just a very brief summary of where that relevance comes from. This is one of the more replicated observations. So these are figures from a paper published about 5 years ago. Essentially, in this study, as well as in all 3 studies that are noted here on the plot, the researchers evaluated the effects of psychedelic administration in patients diagnosed with terminal cancer. So these are patients that are facing an illness that is likely to kill them, and this can be obviously very overwhelming. It can increase symptoms of anxiety and depression. And what these plots show is that in participants after receiving a dose of psychedelic, they show a dramatic decrease in their self-reported anxiety symptoms, depressive symptoms. People tell very touching stories about how their experience with these drugs completely reshapes their perspective on their imminent death in a very powerful way. This has been replicated across these 3 different sites here. So this graph shows evidence for clinical relevance of these drugs as potential therapeutics, in this case for treating associated depressive and anxiety symptoms. More broadly and more recently, these drugs have been applied in the context of treating depression as well as other disorders. I'll focus on depression here. There have been a trio of studies published out of the group out of a research group based in Brazil, showing that ayahuasca, which again uses NN-DMT or has a psychedelic component NN-DMT, evidencing that ayahuasca can treat depressive symptoms. So this started with an open label study in 2015 in a cohort of 6 participants, I believe. And here what you can see is that 1 day, 7 days, 14 and 21 days after administration, depressive symptomatology as measured with these 2 indices is decreased as a function, is decreased following the administration of psilocybin. And its onset is relatively fast. You see it within a few hours of administration and extends out for a couple weeks. They replicated this observation in a larger cohort of about 15 to 20 patients, again in an open label study design. And then more recently in 2019, Fernando Pajana-Fontes showed in a placebo-controlled framework that again ayahuasca was associated with a reduced depressive symptom severity and superior response as well as remission rate compared to placebo control. So evidence that ayahuasca can be a useful therapeutic for major depressive disorder. These findings have been echoed in the context of psilocybin. Perhaps most prominently, this is an open label study done in the mid-2010s from Imperial College London. Robin Carrott-Harris facilitated this study. These are patients with treatment-resistant depression. They participated in an open label trial where they received 2 independent doses of psilocybin. The first one was 10 milligrams. The second one was 25 milligrams. So to give you an idea about that, 25 milligrams is gonna evoke a pretty profound psychedelic experience. 10 milligrams will certainly evoke a perceptual response, but it was really seen as a dose to sort of orient these patients towards the nature of the psychedelic experience. And what they showed is a very acute and dramatic decrease in depressive symptomatology. Again, this is particularly remarkable because these are treatment-resistant depressed individuals, and this antidepressant effect was also maintained over a long period of time. So following this double psychedelic administration in patients, they see decreased depressive symptomatology 6 months after administration, providing some really interesting evidence for this. These clinical effects have been echoed more recently in a double-blind delay to treat protocol with from Johns Hopkins group. This was published in 2020. As well, the same Imperial College London group published a head-to-head protocol where they evaluated the effects of psilocybin administration, this time 2 doses, 25 milligrams with 3 weeks in between, compared to administering just escitalopram, so very standard frontline SSRI pharmacological treatment. What you can see down here in the plot with the red line is the antidepressant effects of psilocybin and escitalopram. And I won't get into the nuance of the details between between the difference between the groups, but suffice it to say, psilocybin itself certainly shows a decrease in depressive symptomatology across both of these studies. So these are all clinical trials that I think underscore the exciting therapeutic potential of psychedelics in various treatment cohorts, and this has been shown across a number of other populations as well. What I'm going to talk about for the rest of this talk is what do these drugs do in the brain? So we have this kind of interesting situation where we have these drugs for which we think there's evidence of clinical efficacy, but we don't necessarily know why. And usually the drug development framework is the other way around. You target a mechanism, you develop a drug that you know hits that mechanism, and then you cross your fingers and you hope it works in the clinic. And we're kind of in a reverse situation here with psychedelics. I mentioned before the role of the serotonin system in mediating the effects of psychedelics. The serotonin system is a system that we can image in humans in vivo with positron emission tomography. It's a system that our research group has spent a lot of time doing over the last 20 to 25 years. We have a very large repository of serotonin PET scans for a number of different targets. Some of them are highlighted here from a paper that we published in 2017. And this includes, among other things, the serotonin 2A receptor. And so I'll talk about how we tried to leverage that expertise with brain imaging of the 2A receptor and trying to understand some of the pharmacology of psychedelics in the living human brain. So the first thing we did is a very basic study. This was a paper we published in 2019, where a PhD student in the lab, Martin Madsen, we created a study where we recruited 8 healthy participants. They received a variety of doses of psilocybin, which are articulated here in this column. They did a PET scan to image the serotonin 2A receptor in the human brain at baseline, so independent of any drug administration. This is with a radioligand that we call SYMB36 or C11 labeled SYMB36. Then on the day of intervention, they come in. It's open label. Everybody knows that they're going to get psilocybin, but they don't know what dose they'll receive. They receive psilocybin in the morning, and this is followed about 45 minutes later by a second SYMB36 PET scan. Now, these PET scans take about 2 hours. During this PET scan, we're asking the participant how they feel, and we're also taking blood samples to measure plasmapsilocin concentrations. And so they're having the psychedelic experience while they're in the PET scanner. In most of the participants, 5 of these 8 participants, we waited a period of a few hours, and then we actually did a second PET scan on the same day. So again, a 2 hours PET scan using the exact same framework. And the basic idea of what we wanted to evaluate is the binding properties of psilocin to the serotonin 2A receptor. So what we expected to happen is this. In a normal PET scan, you have a theoretical cell body like this. You have receptors on the cell membrane, and then you have endogenous neurotransmitter is indexed by these black triangles. It's going to bind to the 2A receptor. When you administer a PET radioligand like SYMB36, it's also going to bind to 2A receptors. So it's got a high affinity and specificity for the 2A receptor. And that the radio label is going to eventually emit a signal that's going to allow us to determine areas of the brain where there are a lot more of these receptors. And so we end up with a map like this, where we have a distribution of 2A receptors throughout the brain, areas where it's relatively high. For the 2A, that's mostly throughout neocortex. Conversely, when we introduce an external pharmacological challenge, like for example, psilocybin, what we expect is it's going to bind to 2A receptors. That's going to decrease the number of receptors available for our PET radioligand to bind to, and that's going to decrease the signal that we're going to get. So we're essentially looking for a delta in our PET signal from the baseline to when drug is on board in order to measure the amount of occupancy of the drug at the 2A receptor. And in fact, this is what we see when we did this study. So on the left-hand side, you're seeing a representation of the distribution of 2A receptors in the neocortex. You can see that it's high at baseline, and there's evidence for a lot of binding when psilocybin is on board here on the right. If we plot that magnitude of occupancy, which we can express as a percentage, if we plot that as a function of how much drug is in the system, and we estimate this binding structure, we can see that there's a very tight relation between the amount of drug in the blood as well as occupancy in the brain, suggesting that if you don't have the luxury of a PET scanner, that you can know a lot about what's going on in the brain after just as a function of how much drug is available in blood. And this is quite nice to see. We expected to see this, but it's really nice to affirm this association. And we estimated an affinity of psilocin for the 2A receptor in the living human brain. We also saw that the amount of dose, the amount of drug that we gave was also nicely related to occupancy, suggesting that, again, we can know a lot about what's going on in the brain in terms of the acute pharmacology with something relatively peripheral like a blood sample or just the amount of drug that we've administered. Yes, so this was a nice demonstration for us of what we could do with PET imaging in humans in vivo to try to understand acute pharmacokinetics of the drug. Another element of how psychedelics work that we were quite interested in was the lasting effects. So we did an independent study of this where we wanted to explore these lasting changes. So I showed some plots indicating that the antidepressant effects are quite maintained. And an outstanding question in the field is, why is that the case? What neurobiologically changes following the administration of a psychedelic that evokes these lasting effects? So we sought to evaluate that with again, a specific focus on the 2A system. So we designed a study which is articulated here. It's an open label study in small study in 10 participants. Everybody does baseline brain imaging as well as some questionnaires. On an independent day, they receive a single dose of psilocybin. They're administered this in essentially a hotel room setting that we have here on the campus. They then return after 1 week to complete, again, an MR scan session where we get bold fMRI data, resting state fMRI data, as well as another SYMB36 PET scan. And then we actually ask them to come back again at 3 months to look at changes again at 3 months. And we did this in part because we were also curious to see about changes in personality measures, and we wanted to evaluate those at 3 months. So when we did this study, this is generally the study design. There were 10 otherwise psychedelic naive volunteers, about 30 years old. They received some varying doses. Most of the studies that we've done have done dosing based on weight adjustment. So the first 4 participants were this 0.2 mg per kg and the last 6 were 0.3. This is the range of the absolute dose. These are doses that evoke a psychedelic experience. If you think about it, I mentioned a 25 milligram dose in some of the clinical studies earlier. That's 0.33 mg per kg for a 75 kilogram person. So these doses are certainly evoke a psychedelic experience. And this is just a couple photographs of the environment of the administration. The first thing that we did was we wanted to say, okay, we have this nice evidence that we have this acute occupancy of the 2A receptor. Do we see a change in serotonin 2A levels from baseline to 1 week after administration? And so here again, you're seeing a cortical rendering of 2A binding. And the top is the baseline average across our participants, and the bottom is the follow-up across our participants. And if you're kind of squinting your eyes to sort of see what areas there are changes in 2A levels, let your eyes relax. At a population level, at least, we see very little evidence that there's a change in brain 2A levels throughout the neocortex. So these 2 bars are the population means. You see some people do go up, some people do go down, but at a population level, there's not really much evidence for a change 1 week after administration, despite the fact that we can see acute binding after drug is administered. This is a bit interesting because we actually showed, or not me, but a PhD student in the lab, David Aitsu, who's actually now he was a clinical psychiatrist on some of the studies at Imperial College London. He showed when he was a PhD student here in Copenhagen that chronic users who are otherwise healthy controls, but individuals who self-report having taken 2A agonists quite a bit, actually show a relatively decreased 2A binding. And that's what you can see here, distinguishing serotonin 2A binding on the left in healthy controls compared to self-reported agonist users. So this contrasts a little bit with our observation where we don't see a change. Perhaps that's a function of the participants, perhaps that's a function of repeated administration. These agonist users often took these drugs multiple times. So exactly what the discrepancy is not clear, and I'll come back to these 2A data in a minute. The last sort of in the context of the same project, we also did resting state functional connectivity. So it sounds like a number of folks here likely have some familiarity with resting state functional connectivity, so I won't bore you with the details. The basic idea is you're lying in an MR scanner. We're doing a bold fMRI sequence while you're not really doing anything in particular. You're letting your brain wander, or you're letting your mind wander, and we want to look at correlation structure in the time series across brain areas. So we estimate some connectivity matrix, some estimate of correlation structure, and this allows us to look at, for example, network connectivity. We can set regions together into groups to define canonical resting state networks like default mode network, dorsal attention, so on and so forth. So in this same cohort of 10 people, we also collected these resting state data, and we wanted to ask, well, is there any evidence for changes in functional connectivity 1 week after psilocybin administration? And we did see some evidence for this. So this is obviously quite a small sample size of 10 participants, but we see at least within the executive control network, we see that connectivity within this network is significantly decreased 1 week following psilocybin administration. So here's just a heat map plot of the individual within network connectivity estimates on the diagonal, and between network connectivity estimates on the off diagonal. This is the one association that was significant after correcting across all these tests. And here you see the estimated ECN connectivity at baseline 1 week after administration and after 3 months. So you can see that after 3 months, it's starting to come back up. It's numerically decreased, but it's not different from baseline. So this change that we see is significant after 1 week, providing perhaps some evidence that part of how these drugs have their effect is through modulation of executive control network communication. But again, this is in the context of resting state in this group of 10 people. We're actually eager to try to test this in a much larger sample, which I'll come back to in a minute when I talk about perspectives. So the next and sort of the last study that I just wanted to talk to you about is a study where we wanted to go back again to looking at the acute effects of psychedelics. And in here we were particularly interested in the acute effects on distributed brain functional connectivity again. So this is a study that we did in a group of 15 healthy participants. Again, this resting state fMRI 10-minute acquisition. You can see the doses, again, this sort of 0.2 and 0.3 mg per kg dosing that we do. And up here at the top, you can see a bit about the study structure. So all participants, this is a single blind crossover study design. I'm only going to talk about the psilocybin data here. On the day that participants receive psilocybin, they complete a scan session before receiving drug. So we get something of a baseline connectivity estimate. They then receive this dose of psilocybin, and then we scan them multiple times after the dosing. So they're spending effectively the entire day at the MR scanner, and we're doing 10-minute scan sessions at multiple time points here, about 40, 80, 130, about 300 minutes after drug administration. So they're having a psychedelic experience in the MR scanner. Certainly comes with its challenges, but we did this with the desire to evaluate the acute effects of psilocybin, but also to relate it to self-reported drug intensity throughout the scan session. We also took blood samples to measure psilocin concentrations to ask whether the magnitude of the effect of drug on resting state connectivity was associated with the amount of drug in plasma, which again we know is related to 2A occupancy and perhaps indicative of the magnitude of the effects. So the basic summary of the effects is that we see that, and this is consistent with what other groups have reported, is that psilocybin generally decreases network segregation. So this is a similar sort of heat map plot on the diagonal from the bottom left to the top right. This describes the associations between the self-reported subjective drug intensity and the magnitude of connectivity. So the basic idea, these blue squares are saying that the more intense the psychedelic experience for participants, the lower the within network connectivity that we see for default mode network, as well as these sort of higher order transmodal networks. And these effects are relatively smaller in the context of more sort of unimodal sensory processing areas. Conversely, when we look at between network connectivity, so connections between regions that belong to separate networks, what we see is that there's a positive relation between the strength of the psychedelic experience, so the self-reported drug intensity, and the magnitude of these between network connectivity estimates. And that's what these off-diagonal warmer colors indicate, these positive associations. So again, this is not, this has been shown by other groups as well, generally, but not in the context of associated with subjective drug intensity in plasma psilocin, is that within network connectivity seems to go down as a function of the psychedelic experience, and between network connectivity goes up, hence that decreased network segregation across participants. And here, these are the effects in a graphical form. This is for the default mode network specifically. This is the average within network connectivity and its association with the magnitude of drug intensity. And here's the average between network connectivity and its association with drug intensity. So averaging across all of these off-diagonal square elements is what you see over here. So again, giving us some indication that the magnitude of the perceptual experience seems to relate to the changes that we see in connectivity structure. We also did some other analyses, including looking at global correlation and kind of consistent with this effect on between network connectivity. We see brain areas involved in regions like executive control network and dorsal attention network, which show a positive relation between their global connectivity score and the magnitude of the experience or the amount of plasma psilocin in the blood. If you imagine these two brain surfaces that you see here, if you imagine taking an axial image through the relatively upper part of the brain, something like this, you can generate what's to date my favorite brain image that we've seen. So this is actually a real data showing areas of the brain whose global connectivity is positively correlated with SDI. So in a way, if you'll indulge the humor, this is your brain on drugs. So that these are some of the acute effects that we've shown with our data in our sample. And so, yeah, and then one other thing I wanted to mention, because I thought perhaps some of you might be interested, we're exploring also some alternative quantifications of the resting state data. So we have an engineer in the lab who's been looking at not just static connectivity, but also time varying functional connectivity using a strategy called LIDA, which I won't go into the details of this as a strategy, but basically the idea is we estimate a connectivity structure at every time point across our scan sessions, and then we do a decomposition of this connectivity structure at every time point, and the resulting eigenvector is 90. It's of length 90 because we're looking across 90 regions. We take all of these length 90 vectors and project them into this 90-dimensional space, do some clustering to estimate brain states to ask whether psilocybin modulates the frequency with which you occupy varying brain states. And there's some there's some assumptions that we have to make. For example, we have no idea how many brain states exist in reality, but we approximate that, and we did some exploration for how the amount of time spent in specific brain states might change as a function of how much drug you have on board. And so these are just some plots evidencing that that there are some specific brain states for which we see the amount of time spent in this state is significantly related to the amount of plasma psilocin across our sort of exploration of how many total brain states exist. And then this plot is extremely busy, so I'll apologize in advance for this, but these are just the two detailing of the two brain states that we observed that seem to show an association such that the more plasma psilocin on board, the less often these frontoparietal brain states are visited by participants. So again, showing that not only do we see effects on static connectivity, but the dynamics of connectivity also seem to change in a meaningful way as a function of how much psilocin is on board. But I'll just kind of leave it at that. And if anybody's interested, I'm happy to come back to it later on. So that was certainly a bit of a whirlwind of some of the research that we've done here over the past couple of years in the context of psychedelics. I think if I'm looking, I've got just a few more minutes to stay within this 40-minute time window. So it's a bit of a whirlwind. I apologize for that. I'm happy to clarify any questions that you have, but I wanted to kind of give you an overview about that. And then with the last few minutes of the time, I just wanted to sort of take a step back and sort of share with you some perspectives that I have about this broader field of psychedelic brain imaging, sort of give you a broader perspective about where it is, and beyond some of the studies that we've done here within our lab. So, oh yeah, and before I do this, sorry, I've mentioned mostly effects of drug on brain imaging measures. We've also been exploring some of the effects of psychedelic administration on personality and other measures. All of our studies have been in healthy controls, so we're looking at personality changes. We replicated a previous observation about Psilocybin-induced increases in trait openness. We also see evidence for decreases in neuroticism, increases in conscientiousness, and also increases in trait mindfulness. So this is from our open label study in 10 participants looking 3 months after administration. We see these changes in self-reported personality measures. We see some evidence that the change in that 2A binding, I told you there was basically no change in 2A binding at a population level, but if we look at the individual difference, it seems to show an association with the magnitude of change in personality. And additionally, we see that 2A binding might show some evidence for an association with the magnitude of the acute psychedelic experience. So people with more higher serotonin 2A levels in the brain seem to have relatively lower mystical experience effects, self-reported. And this change in mindfulness, actually, we've looked now across all of the participants we've brought through. This is about 50 sessions across about 45 people where we show this increase in trait mindfulness across participants. So there's some personality elements of these compounds that we're also exploring and trying to relate to some degree to the brain imaging data. So that's it for the imaging stuff on our side. Now I'll kind of take a step back and talk about some other stuff. So we a couple of years ago, I sat down and wanted to write a review about how psychedelics work in the brain, especially focusing on resting state connectivity. So I did this. I sat down and with a graduate student in our lab, John McCulloch, we sat together and made kind of a table overview of all the psychedelic resting state studies that have been done. And pretty quickly, we saw that we couldn't really do a review because, well, nobody was doing the same thing. So it's kind of hard to aggregate studies that are quantifying completely different things. So instead, we wrote a review basically saying that everybody's doing different things. We should all kind of stop doing that. Otherwise, we're not really going to know what's going on, or we're not going to be able to corroborate each other's findings. There's certainly a lot of utility in the sort of inventiveness that these compounds have inspired in people in terms of various types of quantification, but we kind of need to balance that with sort of replicating theories and models across different data sets. So we published this in a review that's out earlier, came out earlier this year. This is the main table from it. And I want to use this table just as a springboard for giving a perspective on the psychedelic literature as a whole. First of all, this is an overview of how many publications have been derived. This table articulates all the unique data sets at the time of this publication. Green is psilocybin, orange is ayahuasca, blue is LSD. On the far right-hand side, you can see the total number of publications that have been derived from each unique data set. And what stands out to you is undoubtedly is that you have this one study and this one study, which have produced in fact, the majority of publications in the field. And this is articulated down here as well. You have a temporal representation of these studies and sort of spin-off studies that have been published. And this is just to say that the bulk of the field comes from 2 specific data sets and 2 unusual data sets in that they administer their drug intravenously. This is not a criticism of those studies. That's a testament to their data sharing. That's awesome. But it's a bit of a warning that a lot of our perspective on psychedelics is colored by 2 studies, 2 relatively small studies that administer the drug in a way that nobody else does. And these studies were done a long time ago. They had a perfectly good reason for doing it the way that they did. But it's really to say as a field, we kind of need to try to replicate each other's findings, try to validate these models that have emerged over the years. The other point I wanted to make is these studies are relatively small. You can see here the biggest study to date, according to this table, is 24 participants. These studies are hard. They take a lot of money. They take a lot of personnel. They take a long time to do. But nevertheless, we need to think about expanding our sample sizes if we really want to draw firmer conclusions in this space. The other thing I wanted to point out with this table is that to date, most of the studies have focused on acute psychedelic effects, and I've circled here the 3 prominent studies that have looked at persisting effects. That's what this letter P means. There are some other studies you can see that have that P next to them. These are effects that were evaluated about 24 hours after drug administration. So the ones circled really are looking at effects more than 1 day after drug administration. These studies have a sample size of 12, 10, and I think 20, if I remember right, from the Dawes paper. This is to say that there's a lot that we don't know about the neurobiological mechanisms associated with the lasting effects of psychedelics. And this is of great interest for future studies going forward. I'll quickly touch upon this neuritogenesis, synaptogenesis field. So this has grown quite a bit in interest. This is from a study done in 2018, where they looked at cultured neurons and showed that psilocybin or psychedelics increased neuritogenesis and synaptogenesis in these cell cultures, sparking a big interest in the synaptic plasticity mechanism. We showed with a compound called UCBJ, which is a marker for the SV2A protein, that there's actually an upregulation of binding in postmortem tissue in pigs following an intravenous administration in the hippocampus and the prefrontal cortex. The extent to which this mechanism is critical for the psychedelic experience is of interest, and I think largely to be determined. Really showing this in humans is going to be of great interest going forward. And then the other thing is there are some other kind of interesting lingering effects. I mentioned the polypharmacy effect of these drugs, and I think it's just kind of amusing to note that one of the first PET studies done with psychedelics was done way back in 1999 by Franz Wallenweider, where he showed that psilocybin induces a profound increase in release of endogenous dopamine, as indexed by raclopride. In fact,
Hey, sorry, Patrick.
yeah.
We have a meeting at 4:15, so do you think you can maybe wrap it up in like 5 minutes or so?
Yeah, for sure. I have this, I think, in one more slide, and then I'm done.
Okay, perfect.
Yeah, thanks for chiming in. And this is interesting, I think, because we don't think that psilocybin touches the dopamine system. So what's the mechanism mediating this effect is a bit to be determined. I think there's some good reasons for this. I think there are some plausible reasons for this, but it's really just to say that there's a lot about the molecular and functional mechanisms of psychedelics that we don't really understand, and for which there's some intriguing evidence. So that's the last slide. So I'll just wrap up here. I'll say thank you very much. I mentioned previously that these are really difficult. These are time-consuming and personnel-consuming studies to do. So our research team, I'm super thankful to them. Gita Knudsen, who leads our group, and then the pictures of folks here are folks whose data that I showed throughout this presentation. So thank you guys very much for your attention, and I'm happy to take any questions.